Structure and functions of promoters and terminators. A specific length of DNA on one strand of double helix represents a gene. Each gene is divided into two regions, namely coding region and non-coding region. Coding region, also called it as a structural gene region. So this coding region of gene consists of sequence of nucleotides having hereditary information. The coding region directs the synthesis of enzymes or proteins through its transcribed mRNA. The coding region transcribes only one long mRNA molecule through which protein synthesis is carried out. Non-coding region of the gene. Coding region is flanked by non-coding region on its both sides. However, non-coding upstream region, see here you can see non-coding upstream region here. It is organized to form promoter or promoter region. See here promoter, it lies over here. So we will see some details related to the promoter. Promoter gene region, you can call it as a promoter gene region. So this region of gene, it consists of 7 nucleotides in its middle portion. This set of 7 nucleotides is called Pribnow box. See here, Pribnow box. These nucleotides, 7 nucleotide set, it is called as a Pribnow box. It is also called as a Pribnow sequence. This sequence of nucleotides within nucleotide bases in DNA determines the site for initiation of transcription of mRNA by coding region. Then mRNA transcription by coding region or coding sequence takes place in presence of enzyme called RNA polymerase. But the rate of synthesis of RNA polymerase is controlled by promoter. Initially, RNA polymerase is attached to the promoter. Then it moves along the operator or initiator region and ultimately reaches the coding region. When RNA polymerase reaches coding region, then only protein synthesis starts or enzyme synthesis starts. Now initiator. Or initiator region or operator region. See here, this is initiator. So it is a short stretch of few nucleotides between promoter and coding region. Between promoter and coding region. This is promoter, this is coding region, and in between that, this small stretch, that is initiator, is located. And this is called as a initiator site. When RNA polymerase attaches to promoter, RNA polymerase moves along initiator from here to here, moves along initiator and reaches coding region. Reaches coding region. Like this you can show it reaches to coding region. The initiator signals starting of transcription of the coding region for mRNA synthesis. Thus, initiator directs the coding region whether or not the coding region to be repressed by repressor protein to transcribe mRNA by coding region. The initiator, call it as an operator, is closely linked with coding region in the upstream direction. This initiator consists of 27 nucleotides in case of Escherichia coli. Now terminator. See this one, terminator. The non-coding downstream region of gene. Non-coding downstream region of the gene, that is this one, consists of set of nucleotides, and that is called as a terminator, or also called as a regulator gene region. It transcribes its own mRNA on which repressor protein is synthesized. The repressor protein synthesized under direction of terminator, it reaches to initiator, that is operator gene, and it binds with initiator. When repressor protein is bound to initiator, the movement of RNA polymerase from promoter to coding region is blocked by repressor protein, and thus repressor protein stops or terminates the synthesis of protein by mRNA that is transcribed by coding region. In this way, terminator acts as stop signal during protein synthesis. Thus, promoter, initiator, terminator, and coding regions of the gene coordinately work together for a specific function.